In 1961, Nirenberg and his graduate student Matthaei became the first to determine that a specific RNA sequence coded for a specific amino acid. Let's explore how they did it. Nirenberg began preparing extracts from E. coli cells. This extract contained all the components needed for protein synthesis in a free cell system, including tRNA, ribosomes, and amino acids. He blended the bacteria to create almost a bacteria soup composed of cytoplasms and ribosomes. Nirenberg and Matthaei began to experiment with various sources of RNA and even created a synthetic RNA called poly-U. This RNA only contained the nucleotide uracil. First, the scientists added the poly-U into the E. coli extract. Next, they added 19 unlabeled amino acids and a single radioactively labeled amino acid. Then they allowed the test tube to incubate for around 30 minutes or so at 37 degrees Celsius. During this time, translation would take place. At the end of the incubation period, protein precipitate was found at the bottom of the test tube. The scientists filtered out the precipitate while the free amino acids were left behind in the extract. After filtering was complete, Nirenberg and Matthaei tested the filter for radioactivity. They decided to repeat the experiment 20 more times. Every test tube contained essential components for translation in all 20 amino acids. However, each test tube contained a different radioactively labeled amino acid. Next, they added the same RNA homopolymer, poly-U, to each of the 20 tubes in hopes to isolate the resulting polypeptides and test for radioactivity. The results showed that radioactive protein appeared in only one of the test tubes, and that one was the one labeled phenylalanine. This concludes that the RNA codon UUU codes for the amino acid phenylalanine. Through similar experiments using homopolymers, they discovered that the codon AAA encoded for lysine and the codon CCC encoded for proline. Nirenberg began working with another scientist by the name of Leader. In 1964, Nirenberg and Leader developed a technique that used ribosome-bound tRNAs. They discovered that even a short sequence of mRNA, even as short as one codon, would bind to ribosomes. After the mRNA ribosome binding, the codon sequence on the mRNA would base pair with the corresponding anticodon on a tRNA carrying the amino acid for that matching codon. The scientists synthesized short mRNAs with known codes. Then two other key elements were 20 charged tRNAs with attached amino acids. One amino acid would be radioactive. The tRNAs were charged with radioactive phenylalanine. The second was ribosomes. Next, they added the mRNA to a mixture of ribosomes and the tRNAs. The corresponding tRNA anticodon then attached to the matching codon on the ribosome-bound mRNA. After binding, the scientists passed the mixture through a nitrocellulose filter. The filter would catch any attached tRNAs and allow the unbound ones to pass through. Results found that with using the mRNA codon GUU, tRNA with the amino acid valine was caught in the filter. This meant that the codon GUU encoded for valine. Through similar experiments, Nirenberg and Leader cracked 61 different codons and their amino acids.